So thank you very much. So today I'm going to be presenting on a One Health program that we've been developing as part of the AusPathoGen program in Australia. So the AusPathoGen program is the largest pathogen genomic translational research program in Australia that brings together experts and academics from all states and territories across the country and has fantastic representation from public health laboratories and departments of health. The overarching aim of the program is to optimise national genomics-based surveillance of infectious diseases. And this program is underpinned by the AusTracker platform that you heard my colleague Claire present on in the prior session. We have a number of different pathogens within this program, and the E. coli One Health project is one of them. So the project had three separate components. We had a retrospective project where we analysed all of the published and in-house E. coli collections across Australia over the broadest time period that we could account for. And we did this to get a bit of a picture of the diversity of E. coli in Australia, because we have very diverse ecological niches and lots of different contexts that would impact diversity and transmission events. We also used it as an opportunity to start assessing different tools and methodologies for a One Health analysis context, because a lot of the tools that we use in public health contexts aren't necessarily directly applicable to One Health analyses. And whilst we were analysing this retrospective data, we started planning our prospective study, which was the largest component of this project. It was a 12-month snapshot that we conducted just within the state of Victoria, because Australia is a massive country and the state of Victoria alone is larger than most European countries. We sort of had to restrict ourselves geographically initially to start planning this program. And we did this to assess different sequencing strategies across different sample types and really think about the feasibility and utility of routine surveillance for E. coli as a One Health scenario. And the third major component of this program was stakeholder engagement, where we had a lot of focus on workshops and training with all of our stakeholders and our key industry partners. And this was very much led by the industry and the different sectors to facilitate these workshops, because we needed to know what they wanted out of this type of surveillance. So when we came to design the snapshot, we really looked at that retrospective study. We looked at the sectors that had been poorly covered or not included at all to help design sectors to focus on. Sample types that had been missing historically from One Health collections were also a primary focus, and underrepresented groups. Again, we really looked at engagement with stakeholders to ensure that we were meeting the needs, and also that our reporting would be tailored in a format that would be useful to stakeholders and that they could use as actionable outputs. So first off, we designed the strategy to think about all the different sectors that we were going to sample across this 12-month period, how often we would go back to each different sampling opportunity, how many samples we would collect at each point in time. We had a robust laboratory methodology for all of the different sampling types that we collected, and we were doing both Illumina sequencing to begin with, and we will go back and do some ONT sequencing as well. And we've also developed a lot of different pipelines and tools to analyse the data, but also to embed some of the unique visualisation requirements within the AusTracker platform. And this entire project had a governance of three structures. We had the AusPathogen executive, which oversaw the entire AusPathogen program, but then we also had a One Health exec and a One Health working group. And the One Health working group had over 40 members from all different sectors and academic backgrounds. And every single decision that we made, from data governance, ethics, metadata variables, sampling strategies, sampling plans, stakeholder engagement, every single decision was a decision made by committee. We wanted as much perspective as we could get on every single decision that we made. So these tiers of the executives and the working group were really critical to make sure that we were thinking about every question the right way and that we were engaging with every stakeholder in the most appropriate way for their needs, but also for their concerns. We spent a huge amount of time mapping all of the stakeholders that we thought that we were going to be able to engage with for this project, and identify the key people within those different sectors and start thinking about the tailored requirements for sampling of those sectors in terms of data arrangements, MTAs, ethics, and all of those kinds of considerations. And it took over 1.5 years to map out all of these different sectors and players and start getting the agreements in place to actually be able to get the samples into the lab. Some of these different stakeholders, it was an absolute dream. Like, we literally, buckets and buckets of samples were being provided. We couldn't keep up. Others were a little bit difficult. Some very terse conversations, a few people hanging up on me in the middle of phone calls. And it was really tricky. We didn't really know what we were doing, but we kept going back to the working group and kept saying, okay, we're running into problems here. We're not getting some great engagement there. What should we be doing? What are we doing wrong? Is it our communication strategy? Is it our approach? You know, what's the threat? What are they perceiving? And the more that we sat down and talked one-on-one with people, we found they were afraid. They didn't understand what we were doing. There's a really bizarre perception that we hadn't thought about, was that DNA and genomics equals blame to a lot of lay people outside of what we do. And they were thinking, you're going to tell me that I've done something to someone else or to some other ecosystem, and you're going to blame me what's happened. And you've got the evidence, you've got DNA. I've seen CSI, I know that you're going to tell me that I've done this. And then we had, one industry person say to me, you're just going to tell me that I've killed a bunch of koalas. And I'm like, that's not what we're trying to do, but I understand your fear. So once we understood where they were coming from, we could completely change some of the language that we were using when we spoke to these stakeholders and tailor the communications to the level that met their needs and met their concerns. And that helped to some degree. Some sectors we still had a lot of trouble with, but at least, we made some progress. So we sort of started understanding some of the unique barriers and considerations to One Health in the Australian context. But I think this is applicable to most contexts when you're doing One Health. The lack of awareness and understanding around genomics, One Health principles and microbiology are really, really hard for some sectors to understand. There were pathogen-specific barriers. There are certain pathogens that are notifiable in livestock and agriculture, and even though we said we were only looking for E. coli, there was the chance that we could come across other things, or we were asking to create a biobank and potentially come back and sample these, use these samples for other purposes in the future. And so making it very clear what we could and could not do with those samples was critical. And we found that for certain industries, pathogen-specific agreements were the only thing that we could do to alleviate some of their concerns. And it's completely valid that they have these concerns. Fear of community perceptions was a massive thing when we were sampling public areas, swimming pools, parks, community farms, community gardens. They did not want people walking around with falcon tubes, with pipettes, looking like scientists, because again, it was this fear that people would think there was an outbreak. I think the pandemic is still very fresh in people's minds, and they still think that there could be lockdowns, there could be implications for the type of research that we were doing. We also had a lot of problems with anonymity. Groups happy to provide samples as long as it could never be publicly traced back to them. And that comes into some difficulties when it comes to analysing these samples and putting them in context of the rest of the collection. And we've tried where possible to mitigate and find a middle ground to these requests. Our communication strategy evolved over the project, but it was really clear that plain language summaries were critical and having really clear infographics. And usually, as funny and lighthearted and quirky as possible was the way to go. It took an incredible amount of time and resources to build those relationships and build that trust, and it wasn't something that we took lightly. we were really trying to understand what stakeholders wanted, and quite often they didn't know because they didn't understand the full potential of what we were trying to do. It was also really difficult in some circumstances to understand all the legislative and organisational barriers to participation. There'd be certain people that were really keen and they'd work their way up the chain and just hear a hard no, we don't do that. And then obviously all the permits, ethics, MTAs were, their own big set of challenges. And we also really thought a lot about how there was a lot of unmet need for sector-specific training, workshops, engagement on genomics. Even though we'd thought about it from the beginning, we'd really underestimated this need. So it turns out, we had a lot of conversations about what stakeholders in different sectors want. There's a bit of a difficulty in balancing their expectations around what's feasible, around turnaround times for analyses, around tailored feedback to each sector. The realistic, desires that they had were around data privacy and data agreements, some degree of tailored reporting within their sector that's specific to their data, actionable results, and upskilling of their workforce. Some of the unrealistic expectations that we quite often met were point source attribution for outbreaks on farms. This came back at us time and time again, farms wanting this from us. And we just have to keep reminding them this was not the forum for that, and we weren't in a position to be able to do that for them, but we were able to talk to them about some of the tools and things that existed that could help them with those scenarios. We also had some issues around, certain sectors wanting access to all the data in the collection whilst keeping theirs anonymous, and, having some negotiations about whether that's reasonable and what we could do to facilitate a middle ground again. And also, we've also thought a lot about the need for further engagement and consultation with Indigenous communities across Australia and how we use this data in a respectful way for their communities. So in the end, we did a 12-month snapshot and we got an incredible diversity of samples within this study across every sector that we'd hoped to engage with. And we're really, really proud of this, and we're really, especially of our wildlife collection, because in Australia that's something that we're very passionate about, is our native ecosystems. So we collected over 1,600 samples for the biobank, and our strategy was to sample them for E. coli and store up to 20 isolates per sample. We've sequenced over 2,500 isolates to date, and depending on funding, we would have the potential to go back and sequence more. For most samples, we sequenced one colony per sample, but we sequenced all carbapenem resistant and all SBL isolates. For human commensals, because of inherent diversity, we sequenced up to 10 colonies, and the same for environmental samples. We're only very early on in the analysis of this data, but we've got over 300 ST types, and we'll hopefully be producing some outputs really soon describing this cohort. The biggest challenge we had was metadata. We'd thought about this so much from the outset of this study, but even with all of the considerations that we had and the existing structure we had for the AusPathoGen platform around metadata, we still ran into so many complications, and it's been amazing over the last couple of days to hear all the different approaches for metadata that we can now think about how we analyse some of our study in that context, but also how we might improve on our approaches in the future. Because, we had metadata that we wanted but couldn't have, metadata that we have but we have no clue how to use. Like, we had vets fill out pages and pages of history of some of these animals, and it's really valuable because some of these animals were, sick or had different interactions that could be relevant, but we don't yet know how to integrate all of that data together. And there's also some considerations of how much metadata we can realistically put into AusTracker while maintaining it as a reasonable platform that it's all usable in an intuitive way. And so we're playing around with lots of different approaches for that at the moment. So the next steps is, we're really focusing on integrating the genomic data and the metadata within the AusTracker platform and focusing on some of the unique visualization tools that are required for One Health data. We're going back now that we've generated some of the genomic data and really tailoring some of the teaching and training with specific stakeholders. And we're also generating case reports back to each individual sample provider. So over 80 different sample reports are going out with a pretty low-level description of the diversity of their samples and a bit of information about what we've done, so that the stakeholders still feel engaged in the process while they wait for the overall analysis to come, because that's going to take time, but we don't want them to feel like we've just taken their samples and nothing's happening and they're not getting any information. We're looking at scoping some of the national One Health genomic priorities across Australia, and we're looking to repeat the study in 3 years' time. So it just leaves me to thank the sample providers, all the members of the One Health AusPathoGen Program and the One Health Working Group, the members of the One Health Exec, and the postdocs and lab team that were really critical in processing all of these samples across the project, and the AusPathoGen partners as well. Thank you.